Hello, good evening. This is Chari, and we are welcome to ABC Bio. Now we are going to discuss about structure and function of glycoproteins. So these glycoproteins contain oligosaccharide chains, which are called as glycans, which are covalently attached to polypeptide chains. So glycoproteins contain protein backbone and glycan chains. So protein backbone generally widely vary in structure and function depending on the different types of proteins. And glycan chains, these are oligosaccharide chains attached to protein backbone that contain monosaccharides like glucose, galactose, mannose, xylose, glucanac, that is N-acetyl-glucosamine, and N-acetyl-galactosamine. Galnac means N-acetyl-galactosamine, xylose, sialic acid. Sialic acid also known as NANA. NANA means that is N-acetyl-neuramic acid. And these are all the monosaccharides present in the glycan chains. And the glycan chains covalently attached to the polypeptide chains through specific amino acid residues like serine, threonine, tyrosine. Serine, threonine, tyrosine have OH group in their side chains, and asparagine, glutamine have amine group in their side chains. So these form glycoproteins. If you want, you can take a screenshot of it. Thank you. Now we're going to discuss about the next part in structure and function of glycoproteins. That is, in glycoproteins, carbohydrate that is called glycans is less in proportion when compared to the protein content. So glycans are highly branched in the case of glycoproteins. There is one more variation, that is a proteoglycan. Proteoglycan is different from glycoprotein. So in the case of proteoglycans, generally contain more amount of that is carbohydrate and less amount of protein. So that is those are called as proteoglycans. But in the case of glycoproteins, we can see less amount of carbohydrate, more amount of protein. So but here in this case, glycans are highly branched. But in the case of proteoglycans, less amount of protein, more amount of that is carbohydrate content. But here the glycan chains are long linear disaccharide repeating units. So now we are discussing about glycoproteins, right? Glycoproteins are of three types. So O-linked glycoproteins, N-linked glycoproteins, GPI-linked glycoproteins. GPI means that is glycosyl phosphatidylinositol linked glycoproteins. So O-linked glycoproteins and O-linked glycoproteins, here in this case N-linked glycoproteins. So amino acid, in the case of O-linked glycoproteins, amino acid accepting sugar bond is serine, threonine, tyrosine. Either serine or threonine or tyrosine. So these three are OH group containing amino acids, so which accept the sugar bond. So in the case of N-linked glycoproteins, amino acids accepting sugar bond is either asparagine or glutamine. These are amide group containing amino acids. You can remember N-linked. N means here in this case amide bond. So because these have amide bond, here N-linked glycoproteins are found. In the case of here, that is O-linked glycoproteins, because these amino acids contain OH groups that participate in the synthesis of O-linked glycoproteins. So synthesized, the O-linked glycoproteins are synthesized in Golgi apparatus as a part of post-translational modification. So N-linked glycoproteins are synthesized in ER, that is endoplasmic reticulum. It takes place co-translationally. So dolicol pyrophosphate is not involved in the case of O-linked glycoprotein synthesis, and the dolicol pyrophosphate is involved in the synthesis of N-linked glycoproteins. So glycans added in a stepwise manner in the case of O-linked glycoproteins. So glycans added as a block in the case of N-linked glycoprotein synthesis. You can take a screenshot of it. Thank you. So about O-linked glycoproteins. So this is the structure of N-acetyl galactosamine. So here it is N-linked glycoproteins. So it is the structure of the N-acetyl glucosamine. Here in this case you can see asparagine. This is the amino acid in the part of protein, that is asparagine, to which the glycogen that is carbohydrate content is attached. So here in this case serine amino acid, for which the carbohydrate content is attached. Serine is in the part of polypeptide chain. Then GPI-linked glycoproteins, GPI I think you know, that is glycosyl phosphatidylinositol linked glycoproteins. Protein part of GPI-linked glycoproteins attached to cell membrane with the help of GPI. So GPI-linked glycoproteins flexible to move due to the GPI. So for example, you can say acetylcholinesterase and alkaline phosphatase. These are the examples for GPA-linked glycoproteins. And the disease that caused due to the deficiency of GPA-linked glycoproteins is paroxysmal nocturnal hemoglobinuria. Then here it is the structure of GPA-linked glycoproteins. Here in this case you can see this is cell membrane. So the cell membrane is attached with the phosphate, that is a phosphatidylinositol. This group totally is called phosphatidylinositol. So to which glucosamine group is attached. So to which is bound to the mannose. This is mannose. 1 mannose, 2 mannose, 3 mannose. 3 mannose groups are there. Moieties, and here it is phosphate En, that is ethanolamine. It is called PEN. This is totally called ethanolamine. So to which protein is attached. Proteins amino group is in this side, and OH group, COH group is this side. So this is the structure of GPA-linked glycoproteins. So thank you. You can take a screenshot of it. Functions of glycoproteins. Glycoproteins have many roles in our body as receptors, hormones, immune function, and enzymes, transport proteins, lubricant, and structural. They have many several structural roles. So signal transduction. They also participate in signal transduction. So glycoprotein receptors transmit signals from cell's external environment to the interior of the cell and trigger intracellular signaling cascade that regulate many cell processes. Then enzyme activity, they catalyze, that is these glycoproteins catalyze reactions in metabolism, signaling pathways, and in many more other reactions. So example, glycosylase and glycosyltransferase. Next, structural support. They also provide structural support. For example, collagen. It has main role in the extracellular matrix. Then immune response. So antigen, immunoglobulin, that is MHC, that is major histocompatibility. These are all formed from glycoproteins. Then hormones, FSH, that is follicular stimulating hormone, that is thyroid stimulating hormone, luteinizing hormone. These are all formed from glycoproteins. And the protective function, so mucins that are present in mucus also provide protection from pathogens.